Thank you, Professor Yang, for the nice introduction and the invitation. After the three phenomenal talks, I'm very humbled, and I think I will be able to give you some new ideas about what we are doing in terms of ovarian cancer. So I really want to mention about the unmet needs for Asian women. We heard about the non-smoker Asian female for lung cancer, but what about some other diseases for this Asian population? So I want to introduce this disease, ovarian clear cell carcinoma. So this disease is a subtype of ovarian cancer. The reason we need to pay attention to it because it is very endemic in East Asian ladies. When you look at the incidence rate, compare the Caucasian, among all ovarian cancer, at best, you get 15% out of all ovarian cancer. But if you look at East Asia, Japan, Taiwan, South China, Southeast Asia, you will see that we see a lot more of these cases. For example, this is the data for our Taiwan registry that we can get as high as 22% of clear cell carcinoma among all ovarian cancer. So, so this disease has high relevance for Asian ladies, particularly East Asian ladies. And then it is associated with actually a benign disease called endometriosis. This is the data from Taiwan done by Professor Wang Penghui using the National Health Insurance Research Database. So they actually case match with a patient with endometriosis versus without endometriosis, and then follow up from this database match cohort, and then to look into the risk of developing ovarian cancer later in life. So what they found is that patients with confirmed surgical diagnosis of endometriosis actually would have high risk of getting clear cell carcinoma. The adjusted HR is about 7.36, whereas compared to non-clear cell carcinoma, the adjusted HR is basically the same as the background. So this is a disease that could arise from this benign endometriosis. There are many other molecular studies suggested that they could have the same founder mutations in ARI1A. So again, this is something that I think a lot of people probably in this room are not aware of. So there are emerging issues for this disease. The first is why is the incidence high in East Asia? We know about this for 3 decades in our field, but no one could answer why. We have a small study on population SNPs. I am not going to talk about it because the data is not mature yet. But today I wanted to talk about the two other emerging issues: how do we better stratify these patients at risk for treatment. So I want to talk about the immune hot features for early stage clear cell cancer. And the next thing is to be able to stratify patients. We need to understand the degree of heterogeneity of this disease. So, so that is where we utilize a lot of the spatial profiling to understand the tumor biology. So this is a progression of ovarian clear cell from stage 1 to stage 4. So stage 1, the disease confined to one ovary. Stage 2 is confined to the ovary and then the neighboring pelvic organs. Stage 3 is above the pelvis to abdomen. Stage 4, you have the distant metastasis. From many studies from my group previously in Singapore and a lot of colleagues, we have shown that during this disease progression at the gene expression RNA level, there's a switch of molecular subtype from the more epithelial towards the more mesenchymal subtype. And then the immune microenvironment would also switch. So the immune microenvironment would switch from a relatively immune cold in the early diseases to an immune hot to later stage. But the immune hot here is still relative because ovarian cancer in general are cold tumors compared to other diseases such as melanoma. So when I came back to Taiwan in 2019, one thing we decided to start a cohort. So I collaborated with Professor Lin Hongwei and our pathologist, Dr. Lin Wei Zhou in National Taiwan University. We sort of establishes 10-year cohort, early stage clear cell from National Taiwan University. So we have a comprehensive outcome analysis. We gather all the archival samples that we get from pathologies, and then we embark on the multi-omic biomarker discovery. So we do the usual things that everyone is doing, the genomic profiling. In this case, we chose a panel sequencing using the Illumina TSO 500. I won't talk about that, but that data, we've actually identified very interesting fusion transcripts from this cohort. We did immune profiling, and that's how we identified the immune hot subtype. And then because we know that we want to be able, we need to stratify this patient at a clinical setting. So we work very hard to develop clinical assays that pathologists can just use for routine diagnosis. And then we also have some AI machine learning. And then I won't talk about that today. So this is the immune profiling that we did for our cohort for early stage clear cell. The early stage here is stage 1, stage 2 disease. So we took 44 of them case matched, and then we do a bulk nanostring RNA profiling using the pan-cancer immune profiling. Surprisingly, if you look at the heat map, you can look at there are two main categories. One is immune hot, one is immune cold. And when we look at the progression-free and overall survival, surprisingly, we found that the immune hot cases actually have shorter progression-free and then overall survival. And if you look at what are they hot? If you hot for, and if you look at all the immune genes, you can see that in general, you can see you probably cannot see it, it's too small, but you can at least see there are a lot of significant, meaning that they're hot in almost every lineage of the immune cell that you can profile. And so it tells you that the entire tumor bed probably is shifted towards a pro-inflammatory or pro-immune infiltration status. So with this in mind, are we really surprised that the immune hot actually is doing worse because it's counterintuitive compared to a lot of the solid tumors? In fact, it's not, because in another independent cohort that we did when I was in Singapore, we utilized a 3 ethnicity cohort, Singapore, Japan, and UK. We did a similar thing to profile the immune gene expression signature. We also find out if the tumor is categorized as high PD-1 or high CTLA-4 gene subtype. You can see from the survival plot that are also doing more poorly. Another data set from China, published in 2022, they also show that if the clear cells show an immune versus non-immune, the immune ones are doing poorly. So being immune hot for clear cell is really bad for the patient. So how do we identify immune hot tumor features? I think this is a way to segregate cold versus hot tumor. For cold tumors, you can loss of a tumor immunogenicity, antigen presentation is defected, there's the impaired T cell infiltration, trafficking, and then you probably would have heterogeneous microenvironment to turn the tumor cold. But in our way, we want to understand what to turn the tumor hot from the cold, right? So it's the other way around. So like I said, we want to try to understand the immune features for clear cells. So for this, we embark on a trial group translational research study. We started from Taiwan. This is in collaboration with Taiwan Gynae Oncology Group, TGOG, from Taiwan. So we have a Taiwan study as a biomarker validation study, and then we move on to the regional study, which is a TGOG 4002 and EGOT. This is East Asian Gynae Oncology Group, collaborative group, to look for the biomarker for this immune hot. This is probably the simplest study that I've ever done. It's actually to collect FFP sections and stain one IHC, one marker, IHC at a time. So we have 3, so it's not even multiplex, all right? Because probably Laura would know for daily clinical practice, the single-plex IHC, it is still the standard of diagnostic assay. But even that, to collect samples from Korea, Taiwan, and Japan to do uniform staining for people to validate it is not an easy task. So this is ongoing, so I won't be talk about the result today, but this watch this space. This is what we are trying to see whether we can push this to the clinic. And then I'll switch gear to the spatial profiling. I think there are the spatial technology has been, advancing very fast. 2020, we have the Nature Method of the Year, which is spatially resolved transcriptomics. And then in 2024, I think just announced today or yesterday, yeah, like the Nature Method of the Year is spatial proteomics analysis. So essentially, what does that do is we transform the bulk or single cell analysis. We put it back to the tissue, put it back to our, FFP sections that we used to look at H&E. We used to look at, the IHC data, and then we analyze based on either protein or RNA, how this, expression pattern, change over the region, change within the tumor. I think Ram just gave a beautiful talk on that. So I will skip this introduction. So we embark on this study to look into how the spatial profiling could help us further identify the immune hot features of this early stage clear cell. So we took a subset from our collection, and then we utilize the whole slide image instead of making TMAs because the ovarian tumors are generally very big, and we want to have a comprehensive, sort of biased, non-biased, a way to select the region that we profile. And then the technology we chose was the GeoMax protein analysis. We utilize the 18-plex protein core plus 10-plex IO protein. So at total, we can do a 28-plex of protein analysis. So you can see that ovarian clear cell carcinoma usually it could be quite complex within the same patients. You can see from the H&E that you will have histologies such as papillary patterns or the tubulocystic patterns or the solid patterns, and these could coexist within the same section of the same tumor. So we, with this spatial technology, we are able to actually dissect out the tumor segments versus the non-tumor segments. In this case, we call the CD45 positive segment, and then we, based on their distribution pattern, we can determine whether this CD45 segment is tumor infiltrating immune cells or the non-tumor infiltrating immune cells. All right. So how do you see the immune hot feature from the perspective of immune cells? We see that if we segregate from this, early stage versus the late early stage diseases, and then you can see that this figure 1C 1 to 2 stage, that the conclusion is that the tumor infiltrating immune cells, they are more enriched in the macrophage lineage compared to the non-tumor infiltrating immune cells. And then the macrophage lineage markers, the CD68, you can see that actually there's no difference between the tumor infiltrating or the non-tumor infiltrating when the disease progresses more towards the late early stage. And then, but there is a paradoxical shift of the non-tumor infiltrating immune cells from the early to the late early shift from the myeloid to a lymphoid lineage. And then what about the tumor cells next to this immune cells? And then we found that there are some immune mimicry features of these tumor cells. Again, we look at this transition from the early to the early late. You can see that if the tumor cells are sitting next to the tumor infiltrating immune cells in the early stage, they start to take on these immune markers like CD68, CD45, and CD4. And remember I said just now that the tumor immune cells tumor infiltrating immune cells are also high in CD68. So we see this neighborhood effects of this pancytokeratin positive tumor cells go along with their tumor microenvironment. And then we see that the protein signal actually will shift towards the immunomimicry signal will actually also shift towards the progression. And this immunomimicry features of cancer cells, the tumor cells have different flavors, and each of these flavors would detect the different outcomes to our patients. So for example, if the tumor cells, the immunomimicry is going through the granzyme B highly expressed type of tumor cells, the patient usually are doing very well. You can see that this, they don't die at all during our follow-up time. However, if the tumor cells that are, there are fibronectin high, so sort of kind of EMT-like, the tumor cells, and they don't have, they have non-tumor infiltrating immune cell next to it. That means usually the outcome is very bad. So we've been able to kind of utilize this spatial profiling technology, even as low as 20x plex of protein, to come up with construct this map for each individual sections, for each individual patient, to help a pathologist, to help us to stratify the patient for treatments. And then we further expand this cohort to another 10 more patients with their pair 46 FFP section. We select much more AOIs, including 600 over tumor and 400 over immune CD45 segments. And then you can see that, for the CD45 segment compound, you have 4 different flavors of this immune. You have this lymphoid enriched, you have this LAG3 enriched, you have this ECM enriched, and you have this myeloid enriched compartments. And then we've been able to tease out based on whether they are TIIs or non-TIIs, based on their stage progression of how this immune, microenvironment would change. What is very interesting is this: when you look at from stage 1 to stage 4 disease of clear cell, you see this green tumor microenvironment. These are the lymphoid cells. So the stage 2 disease seems to have more lymphoids in the immune, but kind of decrease at stage 3 and stage 4. And then the LAG3 cells tend to be more predominant in the stage 1 disease. So it's something we are also very interested to look into it. And then again, if we put the tumor cell and the immune cells back pair for the neighborhood analysis, that you will see that if the tumor cells are clustered as the cold, like immune cells, you will attract more of the LAG3 positive immune cell next to it. And for the other tumor cells, then you will see a shift in more like myeloid or more lymphoid cells at the neighborhood. Okay, so so what about the advanced clear cell carcinoma? Can we do more to learn the biology? We actually adopted a much humble strategy as the Tracer X study to look into the primary versus metastasis pair, and then to look at the tumor evolution. I won't go into the detail because this has been published in the bioRxiv. Essentially, we took ovarian cancer clear cell patient of their primary ovary versus their multiple metastases. We profile all the whole cells, whole slide, and then we utilize the spatial technology by the ROI-based or the array-based or the single cell-based transcriptomic analysis to look at whether we can identify some niche or some unique cells. I would just talk about this COSMEX. This is the single cell spatial transcriptomy. We took one ovary and her peritoneal metastasis and her colonic metastasis, and we profile up to 150 FOVs and then up to 80,000 cells. And you can look into this UMAP that we can identify different clusters of single cells, and then we can annotate them best based on whether there's cancer cell, immune cell, or the stromal components. Just look at the cancer cell. There are 9 different populations of cancer cells. If you look at their distribution among ovary, peritoneum, and colon, just by looking at the color, you can see an organ-specific distribution of different cancer clones. With the help of our pathologists, which help us to annotate which FOV, where they are situated at the tumor center or tumor periphery or at the invasion or at near the necrotic regions, we can see that focus on the pink cell, which is the cancer F, that becomes, it was a small population in the ovary, but becomes a dominant population in the peritoneum. And then if you look at the purple cell population, it was, only present in the invasion and stromal part of the peritoneum, but becomes a dominant population in colon. So I will skip that. We could utilize the trajectory analysis to look at how these cells evolved among these different metastasis trajectories. And then the conclusion was the pink cells that you see as a dominant clone in peritoneum, they're probably the founder cells. And from the gene expression signature, you can see that they're enriched in some sort of the stem cell-like markers. And then when we map them back to the single cell space, you can see that this cancer F, these pink cells, these founder clones, they indeed have a shorter pseudo time at the trajectory, suggesting that they are the older clones, they are the founder origins. And then if we put them up next to, if you see this cluster, which is a founder clone, which you see that is a very tight, very short pseudo time, a neighboring region just next to it, you see a boom, kind of flowering increase of the pseudo time, suggesting that even within a very short spatial distance of the 2 niches, you would have a very protected niche for the founders where you have the daughter clones that are just spreading right next to it. I'll skip this section. So the ultimate vision is really to bring the spatial technology to the clinic so we can have this translation tool that can help us in the clinic. And then lastly, we want to share this last example is a real world case that there's a 38-year-old clear cell patient, stage 4B, extensive disease, and the surgeon couldn't do complete debulking. So she has a lot of residual diseases. She only had partial response to chemotherapy. We profile her genomic alterations, TMB low, MSS, and GI is low, meaning that we cannot prescribe her IO or even PARP inhibitor. She has this MET fusion transcript being detected. So in theory, we can treat her with MET inhibitor. But we were thinking whether we can utilize the spatial analysis to help us find anything to help her. And then when we look into this, her tumor microenvironment, you can appreciate that her bladder metastasis and her some part of her ovarian tumor, in the tumor microenvironment, we see this upregulation of Wnt pathway. Like Ram just alluded to you that this could be a very important immunosuppressive pathway, but unfortunately there is no Wnt approved inhibitors that we could use. So can we do more to for patients like her to help to improve the outcome? So this is just happening yesterday. So this is from National Taiwan University Cancer Center. We just signed a MOU with 10x Genomics to have this first in the world center of excellence of single cell and spatial omics center. So watch this space. Hope we will really bring a change to the patient care. Thank you very much.
Professor Huang's very nice talk. Is there any question from the audience? I was wondering actually, so do you think there is different roles for the different immune populations as you go from stage 1 to stage 4? Because initially it seems less lymphocytes, but they have very high LAG3, so would be more suppressed. but then the LAG3 appears to be going down, but then you also have more. So are they playing, different roles at different stages of initiation and then progression?
It's a good question because we know from the RNA, the gene expression subtypes, stage 1, stage 2, they're probably more epithelial. Stage 3, stage 4, they're more mesenchymal from the bulk. So what we're thinking is that probably there could be a transition, but what I think there's another possibility that stage 3, stage 4, they're probably completely different disease. Okay, from stage 1, stage 2. So the stage 2 is a very interesting transition point. You can see that is a significant decrease of the LAG3 and then increase of the lymphocytes in the lymphoid and then decrease all afterwards. So this, I think you're right. What we haven't looked into it is exactly how these different niches during that transition would tell us. Yeah. And then what is actually regulating that transition. So we think stage 2 probably is the key of this hot and cold. And then from the hot, probably it turns out into bad hot. So the early hot, immune hot probably is a good hot that you can still target it. But then, because you deplete the lymphoids, there are actually no lymphoid cells for you to target anyway. So, but stage 2 diseases are rare. We have a lot of stage 1, some stage 3, stage 4, stage 2, sometimes it's opportunistic. Yeah.
When you have so many different clusters that are molecularly defined, you had 8 different cancer clusters, how do you know which are truly functionally distinct in a way that kind of matters for the tumor progression versus the ones that have some molecular differences but maybe behave similarly?
That is a good question. We've been thinking about that. It's difficult to do functional validation for that because it's almost impossible to simulate, to model each clone. So what we are trying to do is look into the hallmarks, the changes of the hallmarks, and then some the cell interaction, the cell communication from the cell chat, and then to actually help us to pinpoint which subcluster would be the one to go, right? So the cancer F, the pink clone, that is the one that we focus on. And then we have other known clear cell signatures that we can utilize that as our hallmark. And then to see how these clusters are enriched or depleted in those known signatures. By using that, we can further tease out, from that population, whether there are another, more rare population that would be the one to go. Yeah, but that's the best we can do so far.
Yeah. Because of the time limit, I would like to close the section here and let's thank Professor Huang again.